This is just showing you the difference in mitosis and meiosis. Mitosis we really don't care about. I mean, it's important, however. But for these kinds of genetics, meiosis is where all the action is going to happen. And as you can see in mitosis, it's pretty much just cloning, right? One cell, the chromosomes line up and they duplicate, and that's the end of it. But in meiosis, we don't have the full chromosomal package, so to speak, in the daughter cells. So a parent cell is going to produce 4 daughter cells in this case. And because there are 2 divisions, division 1 and division 2, the daughter cells typically, hopefully, would only have half of the genetic material. So in each of these daughter cells, you would only have 23 chromosomes, which makes sense because you don't want to have 46 in each of your daughter cells and then have it fertilized by a sperm. If this is an egg, it's fertilized by a sperm, and then have double, the amount of chromosomes that a normal person has. That's not what a twin is, right? So that doesn't make a twin. You need half of the chromosomes, and then when it mates or is fertilized, you have the full set of 46. But this is the interesting thing, is this is sometimes where issues can occur that, and again, you were right, and that's why it's considered genetic, but not hereditary. There's a process here, number 1, called crossing over. How many people are familiar with crossing over? Okay, so crossing over is just where these chromosomes start to line up. They duplicate each other, they line up, and some of the material is exchanged. And as you can see on this slide, the material from these 2 pairs is exchanged so that they are no longer identical anymore. They share some of this DNA material. Coupled with the fact that once they have the second division, it's random which one of these goes into the daughter cell. So coupled with this crossing over and the random allocation of the chromosomes into the daughter cells, you can come up with billions of possible combinations of DNA, which is why we are all so different. And DNA is such a wonderful thing because although we are so different, we share roughly 99.8% of our DNA. It's only that less than 1% that gives rise to individual differences. That's mind-blowing. You tend to think that variations in the population, in a more logical thing, you think, okay, most of us would share maybe half, right? It's not. It's that similar, that it's less than 1% that makes up the individual differences. Chimpanzees, for instance, we share about 98% of our DNA with chimpanzees. I mean the genome sequence. I don't mean they're actual, that they're like our twin. I don't mean that kind of 100%. But nonetheless, we are most often quite different because of this process. There's actually a permutation you can work out. It would be 2 to the 23rd times 2 to the 23rd, which would result in about 8.4 billion times 8.4 billion combinations. So it's amazing that any of us, even have we have one twin in the world. It's amazing that that could even happen with, the unimaginable permutations that can occur through this random process. So sometimes during this particular event, things can go awry, and that's what I'm going to talk about for some of these disorders that we'll be discussing, if not today, then on Wednesday. So again, this is meiosis that we care about. This next slide is bigger, and I like it because it just shows more simplistically what we're talking about here with the crossing over process and how things can happen during division. Keeping in mind that there are 2 divisions, not 1. This just shows the 1 division, but I'm more interested in what's happening here to explain these mutations. So during this crossing over, there can be some rearrangement of a genetic sequence, and again, that's that trinucleotide repeat of the codons or the CAG or whatever the case may be, whatever the gene is, and that kind of mixing up can give rise sometimes to mutations. And mutations are meant in the world, of course, to sometimes be a good thing, right? We all carry at least 4 that can be lethal. And we all have at least 4 of these mutations in us. So, like I said before, we are mutants. But we don't show it in our phenotype because most of them, luckily for us, are recessive, right? Now, the only reason that we have mutations is to ensure survival. So these mutations are going to make, it's really nature's insurance policy that if something were to go wrong, that some of us would survive, right? We'll be talking about sickle cell today, and that's one of these mutations where people have survived malaria due to having a mutation, which is, again, giving rise to sickle cell disease. So, again, I can anticipate things happening in the future, especially major changes in our environment, where a mutation in either lung capacity or oxygen, delivery in the lungs, or even pigment in the skin, may be beneficial for those people as we start to experience real drastic changes in our climate. So not all of us will survive. Those who have mutations may give rise to, a better survival rate. So this is, again, the idea here. Now, the other thing that's interesting here is that each one of these chromosomes is going to go into, now we're only showing 2. This would happen with all 46. But just for simplicity, we're showing just one pair here. So one of them, of course, ends up in the daughter cell, or all 23. But sometimes things can go wrong in this division, either at division 1 or division 2, where there may be an absence of one of these chromosomes. One of them doesn't go into the daughter cell. This can sometimes happen with the sex chromosomes. So you may only get one X if you're a girl, and none on the other one, in which case it would be XO, right? O meaning lack of a second chromosome. Or 2 might go into the daughter cell, and then when it's fertilized, you end up with 3 sex chromosomes. It could be XYY, XXY, XXX, right? So all these different combinations can be due to problems in meiotic division. So this is why I say it's not hereditary, but it is genetic. It's genetic because it involves genes, but it's not hereditary. It's not a gene being passed on that causes this. It's due to an environmental event. So this is the difference between genes and environment. This is environmental, but it just involves genetics. So this is just an abnormality in the meiotic division. It can happen during zygotic formation in the womb. So after it has already been fertilized. This can occur during the division. So again, in trisomy 21, a particular child may get 2 of pair 21 going into the daughter cell. So rather than getting 1 chromosome 21, the daughter cell gets 2 chromosome 21. And if that happens to be the egg that gets fertilized, or the sperm, it can happen either way. We tend to think it's all females. It can also be due to males, less likely, but it can happen. But if this particular gamete does get fertilized and the other gamete contributes 1, you're going to have 3 chromosome 21, right? Which, of course, is going to be Down syndrome. This is called nondisjunction. And nondisjunction is just when the chromosome abnormally goes into a daughter cell, either an extra one or none at all goes in. So you only have 1 when it is fertilized in the case of the sex chromosomes. So in the case of Down syndrome, 97, 98% is due to nondisjunction, where this fails to split and 2 of them go into the daughter cell, and then if it is fertilized, then of course you would have trisomy 21. The other 2% can be genetic, and this is called translocation. You ever hear translocation? What is it? Are you confident enough to say no? Okay. Okay, translocation is genetic, and the person is essentially a carrier. So they may have part of chromosome 21. Material on chromosome 21 is on another chromosome. It's stuck on another chromosome. It's called a balanced translocation because you don't have an extra chromosome. Just part of chromosome 21 is now on another one. And if that's the case, then you could pass that on to a child. It's very rare, right? Most are due to this nondisjunction, problems in meiotic division. The other 1% are mosaicism, where some of the cells are okay, some of them are not. So some cells have 321, some have normal. And this can happen with Turner syndrome as well. Where a girl is born with 1X. Some cells may be okay, some cells are not, and that's called mosaicism. And that's not genetic either, or hereditary. So again, most of these are due to problems in meiotic division or non-disjunction. Okay? Are we good?